All right, I see people are trickling in. I think we can begin our final session of the DREAM meeting today, discussing the RareX Xcelerate Rare Challenge. So I'm Jake Albrecht, Director of Challenges and Benchmarking at Sage Bionetworks. I help oversee a lot of the DREAM challenges that Sage supports, and so you see my name in a lot of the meetings, so it's great to see the people who participate in our challenges and learn from their experiences as we continue to make DREAM more accessible and easier for people to get involved, to expand the reach and really bring in more people to into the challenge ecosystem. So we've seen a great number of talks today covering a wide variety of the things that DREAM does. Today I will be presenting an overview of the RareX Rare Disease Challenge. So this is a challenge that we had run earlier this year with a new partner called RareX. So a bit of overview on the RareX Data Science Challenge. We had partnered with rarex.org. It's a nonprofit that works with a number of different patient advocacy groups around rare diseases. And so they partner with patient groups to collect patient research and onboard people into their data platform, which currently consists mainly of survey data and genetic test results that they go through and curate. Let's see. So patients who are onboarded to their system, they're interested in sharing their data with RareX. They want their stories heard, they want their data used, they want to advance research and connect with researchers in the biomedical space. And so RareX came to us with a few goals around this idea of a challenge. So the first was to accelerate this collaborative use of data shared by the patients for this research objectives. They wanted to fuel new insights and hypotheses around some of the data sets, and they work with a number of pharmaceutical companies. And so the idea of creating some therapeutic development targets from this challenge was kind of a long shot, but it was something that they were really interested in starting the discussion around. So the RareX data analysis platform is a data set that exists on Terra that they wanted to draw attention to and bring people onto that platform for general research uses. And they also wanted to see what they can use their data for. So come up with some new use cases around the data that they have on RareX. So we partnered with them to help formulate the challenge tasks. And then longer term, they want to do more data challenges in the future. You saw from Paul's presentation that we'll be doing a second challenge next year. And so hopefully that trend continues and we're able to run more of these rare disease challenges. And they also want to extend their network of data partners. And so there's a number of people in the space. We partnered with a few external groups to put together the rare disease data, learned a lot along the way, and want to take those lessons and into the future. And we also want to bring in new patients and advocacy groups into the collaboration and run future challenges with data that they are able to donate. So the focus for the 2023 challenge was on rare pediatric neurodevelopmental disorders. And so these are often genetically defined diseases that have an onset early in life for kids that can lead to developmental delays, intellectual disabilities, a number of other symptoms. And they wanted to understand how these phenotypes can be correlated with the genetic diagnosis and if there was some commonalities around those diseases. And so they had this nice figure on the open science life cycle, starting from the collection to the analysis to creating a data challenge. I love to see data challenges as a key spoke in this wheel of the life cycle, and then that challenge leading into the utilization of knowledge and to fuel future hypotheses. So the tasks that we ended up selecting were to identify previously unrecognized phenotypes. And so for a lot of rare diseases, there's not a lot of literature around them. And so we wanted to help patients who are newly diagnosed understand what phenotypes or symptoms their child may experience with these diseases. There's not a great deal that's well documented. A lot of the cases that are in, say, gene reviews are only a few dozen of patients, whereas RareX has collected data from thousands of patients and hundreds of patients with a single disease. So it's a really rich data set that's has yet to be utilized for these types of phenotypic descriptions of the disorders. So we also wanted to see if there was a way to correlate some of the phenotypes that we had with the genetic diagnosis. This was kind of a long shot. This is basically predicting the genetic disease without the use of a genetic test. And so just looking at the cluster of the phenotypes, how well do those correlate to a single disease? And so this was an interesting question. It was one that worked well with a quantitative metric. And so this was the most quantitative of the challenge tasks that we had. And the third was more of a stretch. So how can we generate new insights for advancing therapeutic research? So how can we take what we know about these genetic disorders and the gene networks and correlate that to, or use that to identify possibly new targets? And some of the goals for the outcomes for this for researchers would be the ability to share some of their rare disease insights, introduction to some of the grant-making organizations who could possibly fund some defined research objectives, and then also present to the RareX scientific board and make connections through their network, which is pretty well developed. And so thinking about why should people participate, this is something that we ask ourselves a lot in creating a challenge, thinking of a target challenge participant. So for researchers, there's the opportunity for working with this new set of data. This has never been released before. There's a lot of underreported diseases and phenotypes that the RareX team has included in their surveys that may not make it to the literature. So this is something that they wanted to share with researchers. And RareX has a objective of getting new researchers onto their platform and finding ways to share their data and make it more accessible. They're all about disseminating that open data set. And importantly, for patients and families, this is something that's it's an opportunity for them to have their experiences heard and researched by data scientists and researchers, and also get a better understanding of how disease phenotypes and outcomes can happen for these pediatric patients who the families are caring for. And so the great irony of rare diseases is that while individual rare diseases are uncommon, the number of rare diseases is quite large. And so there are actually a lot of people who are affected by rare diseases. So I was just curious for folks in the room, show of hands, do you know of anyone who's affected by pediatric neurodevelopmental disorders? We have a few. Actually, everyone should raise your hand because you all know me, and my daughter has a pediatric neurodevelopmental disorder. And so this is something that she's 3 years old now, and going through the process of getting the diagnosis, which takes a long time because you can't do inpatient genetic tests and have insurance pay for it. But there's just a tremendous amount of uncertainty around how you would be able to plan for the future and know what to expect in terms of symptoms. So this is something that's really important for me. It's one of my motivations for pushing forward with the rare disease challenge. So it's really great to be able to participate in this type of research and help understand some of the things you can read the literature and understand things about developmental delay, but there's other symptoms and phenotypes that really affect your quality of life. There's sleep disorders, gastrointestinal disorders that are also correlated with these types of neurodevelopmental diseases. So back to the challenge development. So the RareX organization had collected a large data set for 30 plus rare diseases using their survey, which is the PRO measures here, and the genetic testing results. They also had a subset of the rare disease communities that had more information about the lab values and other natural history data. And they also worked with, they were really proactive in reaching out to some of these other data organizations like CORDS, which is a large open rare disease data set that has some semi-structured data. It's not as well controlled as the RareX data, and also Invitae, or now Citizen now Citizen, that has this much more detailed curated EMR data, but it's much more tightly controlled, and so it was quite a negotiation to get access to those data sets. And so to make the connection between the sets, we use DataVant to tokenize the patient IDs. And here's an example of comparing the RareX data set to the CORDS data, where there's an overlap. 55 patients with Malon syndrome are in both sets of data, so we can merge those records and fill in some of the RareX survey data using some of the information collected in CORDS and vice versa. This is one way to expand the data set. For the challenge itself, when we had set it up, we ran the main leaderboard round over the summer, starting at the pre-registration in May, and then the final round was involved. After the final round, there was a lengthy delay. There was a lot of judging for some of the challenge tasks. And ultimately, the winners were announced in February. And so the data set itself is really difficult to describe in a 30-minute presentation. The data that's been collected involves a lot of survey data. So there's the structure was a general health survey for all patients, and then depending on the answers, they would go into some level 2 surveys. And so if you're familiar with any of these, there's like the Vineland survey is on developmental disorders. There's the pediatric children's sleep habits questionnaire, aberrant behavior for autism and other behavioral disorders, eating assessment tools, disability assessment scales, et cetera. And they also surveyed a number of organ and physiological systems for various health issues that they could anticipate. But ultimately, this means that if you were to take all this data, which we had access to, sort of flatten it into a single table, you would have close to 2,000 patients, 13,000 coded measures, but it's very sparse, and so there's not a lot of information captured for all the patients, because not all patients had skin disorders that required answering that separate survey. So there was kind of a sparse data, but a lot of patient and other details that we had captured. And so this kind of informed some of the challenge questions that we wanted to include. So the first task that we had, just present a quick overview of the challenge results. The data we had used was the 1,400 patients. Half of that was used for training, and here we provided the for a lot of the diseases, 27 diseases, and 223 symptoms that were identified in the survey. So this could be difficulty standing. It could be other sleep disorder scales. And participants were also encouraged to grab any external data. So the goal was to identify some of these under-recognized symptoms and expand some of the definition of rare diseases. So tools like OrphaNet will have various frequencies for different phenotypes, but those lists are incomplete or may not be well aligned with the RareX survey data. We may be getting some different responses. so we wanted to make this as accessible as possible. So we didn't use Docker containers. This was just strictly a code submission that we had would run and verify on the set of withheld data. There's no ground truth, so we're just interested in reproducibility and the significance and quality of the write-up for this task submission. So we did see some great uses of external data for the different approaches that were submitted. We saw a few that you would expect. If you're a statistician, you'd be reaching for your Fisher exact test for this task. There was a really nice use of TF-IDF, which I didn't expect to be used in this challenge. And then there's more advanced tools, more recent, like multi-X rank. We'll hear about later on for the top teams. And so all the solutions were very simple. They were easy to take and implement, and ideally we could use this for future diseases in the RareX database to help curate a set of phenotype frequencies for different new disorders. And so to announce the top teams for Task 1, the RareX organizers did a nice job of identifying what they liked about the top methods and creating different awards. So the best approach of working with RareX and external data was the 3 billion team. You'll hear from later on today. The best open source method for rare disease research. So the Chong Lab at the University of Washington used some easy, accessible tools like TF-IDF in their approach. And then the most innovative approach you also hear from later today was from the systems biomedicine team at Marseille. For task 2, the goal here was to predict the diagnosis based on the symptoms, age of onset, severity of the disease, and quality of life assessments. And so we would provide that information, but to get access to that and the rare disease platform required a full DPA and agreement. So we only shared a synthetic data set similar to the PD1 challenge, where it's just a bunch of tables. It has generally the data types that you can use, and your goal is to perform feature engineering, find novel ways to combine different features, and then because we retrain the models on the new data, the real data, once you submit your Docker model, you can do things like include like an AutoML type of pipeline in your method, or a tree-based approach that works well for general purpose learning. And so this goal was to bring in data scientists and machine learning specialists to work with the data set. And our metric was just the top one accuracy. So how accurately could you predict the one of 16 diseases given the feature sets provided? And we also provided a reference solution, and it wasn't a softball, like something that was easy to beat. This was something that we built using AutoML. So we thought it would be pretty well suited for the task, but many teams beat that method handily. Although they were able to achieve over 50% accuracy, this still means that half the time you're going to get a wrong diagnosis if this is your only diagnostic tool. It's much better than like a 6% accuracy is what you would expect with a truly random guess. So there is a lot of signal there, but it's not something that could be used, and it just highlights the importance of genetic screening. So genetic screeners have good job security on this, but we still have to rely on those tests for definitive diagnoses. So to show the results for the task 2 winning teams, so team Dean Ambit what took top honors. The overall accuracy was above 50%, and using our bootstrap and Bayes factor approach to identify significant differences between teams. You see that the next two teams, the Berger Lab and Guan Lab, were essentially tied for second place. So this was really great to see these 5 teams beating our reference solution that we had provided. And looking at the top 3 methods, ensemble approaches were the most common. The XGBoost and Light GBM tools were used. And although that number of 50% accuracy for in aggregate is not great. You can scan look at the by disease accuracy, and you do see for one in particular, the Wiedemann-Steiner syndrome, there's over 80% accuracy in identifying the disease, while others like DRK1A and CHAMP1 were really not much better than chance, and those are also some of the least abundant cases in the data set. But why was WSS so easy to predict? And so we did a deeper dive into the method that Dean Ambit submitted. You can do a confusion matrix to see how well the different diseases are called. And so you see a few that, like FOXP1 is often predicted. Maybe it's because it overlaps with a lot of the other diseases, even though it was not as accurate. And we can also do a deep dive into why this yellow square was, but why the model was picking up on that made it so accurate. So you can also do a SHAP analysis and look at the relative importance of different features. And so some of the features that pop up are the age that the patient had first concerns about development. There's also some really specific phenotypes like short stature, issues with teeth and mouth, flexible joints, and some other phenotypes that are unique to WSS. And so this disease in particular was easy to pick up from some of the clinical outcomes. And so for task 3, this was the most open-ended of the tasks. Here the goal was to propose or refute a therapeutic hypothesis for any of the rare diseases in the RareX data set. And so here we opened up all the data plus an analysis that was performed by a partner, Netromark, which is a company specializing in AI for patient subpopulation identification. And so they have some really fancy causal modeling approaches for diseases. Their analysis identified immune dysfunction as a potential cause for some of the neurodevelopmental disorders. So what we had provided that analysis along with the full set of rare data, and so this was geared more for clinical researchers who can sign those DPA agreements, which did limit participation, but it did bring people on board to the platform. The metric was strictly based off of judging. So RareX organized a panel of judges to go through and analyze the write-ups and get assess the top teams. So the top teams that came out of that were the Medford Lab at St. Jude for using the best use of the rare disease data set. So this is a group that studies CHD2. They proposed some further research using the RareX data. And then there was another award for most novel approach for therapeutic research that you'll hear from the Guan Lab later on in this session. So that sort of tees up the RareX outcome, the challenge, and some of the outcomes that we had. I wanted to thank the organizing team. There was a lot of people involved behind the scenes from Global Genes / RareX who had set this up. The data providers at Accords and Citizen, and my team at Sage Bionetworks, who had put together the site and managed a lot of the data science activities that RareX had proposed. They don't have a deep data science team, and so we helped develop some of the challenge code for them. And so just some last thoughts. So the outcomes and outlook for the RareX. This was a first for Sage with rare diseases and for RareX. We learned a lot of lessons in some of the processing and standardization, especially with these third parties. We received a great number of hypotheses from the community. It was really, this is why we do Dream Challenges, is to get that diversity of thought in solving a common problem. And there were a large number of the methods that were submitted were easy to implement and run. Our site has helped develop our post-challenge analysis capabilities. And we'll take the findings. There were the task 1 results in particular. There's so many new phenotypes discovered for all these diseases. We can't go through them all, but this is something that I hope will change how rare diseases are shared with the broader community. So with that, I want to thank you for your attention, and we'll take any questions you might have. Or we can, I guess, if there's no other questions, we can bring up our next speaker from 3 billion. Oh, yeah, Pablo.
So I know you're doing a second set of challenges around this, right? So can you just frame on how you're going to build up on this?
Right. So this challenge set was run on the pediatric neurodevelopmental disorders, which is just a subset of the rare diseases. So they'll be looking at other diseases that's still TBD right now. They're working with different data partners, but it will be rare diseases, different target population.
And do they have plans on trying to amplify the cohorts for having more data? Besides also
yes, yeah.
That was the main limitation here, right? And I mean, it was kind of interesting that you bundle a bunch of, I guess they have similar phenotypes initially, so that's what makes sense. But at the end, they're rare. You don't have enough probably genetic information, right? So it seems that that's the limiting factor here. Right? So,
right.
Are there plans? I mean, how fast do getting this data happens and these type of things? How do you see the progress around that part?
Oh, right. Yeah. So with they have a number of rare diseases in hand at RareX. I know Carmen is busy working to organize other groups for their rare disease research, and she has connections with different patient organizations. And so it is always a challenge trying to get new cohorts, but we may see another theme. It could be neuromuscular disorders or other types of common genetic diseases that we could also lump together to get a bit better signal. Yep.
Nice work. Have you considered using some of the other data modalities that can be relevant? For example, some of the epigenetic signatures that can be included. So I know at least one method. I'm not working on the rare disease, but I know, for example, EpiSign is one of the methods that use the methylation data to classify patients with a rare disease. That's my first question. And the second question, so there are some other networks, such as Care for Rare, for example, that try to connect people all over the world. Because for the rare disease, the numbers is low, but when you consider it for the whole world, then the number of numbers. Have you considered to getting them involved in any way?
Oh, that's a great question. Yeah. So the two questions were considering other genetic markers like epigenetic, and also connecting with other organizations like you said, Care for Rare. Yeah. So that's a new one by me, but I think that could be a good resource. Like reaching worldwide is something that is a priority for the rare disease community. I know in my parent Facebook group, like there are people from around the world sharing different stories. So, and with the rare disease, with the RareX data, I think there were about 50-50 US and Europe, but there's no reason why it can't be more global. Yeah.